probably missed a lot. There's a lot to say.
Hello, everybody. So if you cannot hear me on the back, you let me know. No, even later on the talk. So as Chloe said, I'm in the Department of Entomology at Texas A&M. And my main work is on transmission of plant pathogens by insects. This is not the title that I gave. I don't know exactly what was the title, but I changed it. So it's easier. But the talk is the same, don't worry. And so I'm going to be talking about disrupting the transmission of plant pathogens, and I call it a triptych view. Triptych, you know, is the paintings that have 3 parts. And so people in my, I hope that, okay, good. There's messages coming up. So the people who do my job, we like to show this kind of slides in which we call the disease triangle. And in this disease triangle, in fact, we have susceptible, if you have a susceptible plant, I'm going to point with the mouse for the people in Zoom. So if you have a susceptible plant, a competent vector, and a virulent pathogen, and obviously if the conditions, the abiotic conditions are adapted, you may have a disease, okay? So we need the perfect storm for it to happen. The issue is that it happens pretty often, and it's detrimental for us that we want to feed. The important thing in this disease triangle are in fact the adjectives that I use, because if you do not have a susceptible host or a competent vector or a virulent pathogen, in that case, there's no disease, right? And we don't care anymore. And in fact, host plant resistance is the best way for us to protect our crops, but there is not always present. So before going, this is a pathosystem that we're going to be talking about, but before I introduce it, forget about it. I want to give the overview of my talk, so you guys know what we are going to be seeing, because you are not going to see this slide ever again. So you know ahead of time where we are heading. So I'm going to start giving a little bit of history about the pathosystem, so now you are going to be all experts in the disease that I work with. And then we are going to talk about the interaction about LSO and the insect, LSO and the plant, and the insect and the plant. And I forgot to say that why we do that, because if in this disease triangle we're able to disrupt any part of the disease triangle, then there's no more disease. So what we are interested is in disrupting those interactions. So let's go back in time. We are in the 1990s in Saltillo, Mexico, here, very close to the border with Texas. So in this potato field, there's a new disease that is observed and is described. So this disease has some aerial symptoms that are very common to many potato diseases, and these include chlorosis, rolling of the leaves, stunting, so the plants are shorter, decreased vigor, early death. But that, I'm describing many potato diseases. That is not unique. What is unique are the symptoms that develop on the tubers of the plant. So here we have a tuber from a healthy plant and a tuber from an infected plant. The first thing that you can already observe is the difference in size. Okay, so the disease has a fitness cost for the plant, yield is reduced. The other thing that you may see in some varieties are these stripings. This is not soil. That is the tubers that have a discoloration. That is not observable in all potato varieties, so just in a few of them. But what is observable in all potato varieties is when the tubers, that is the part that we eat, are cut and fried, when we make chips, they develop this striping. And because of that striping, the disease was called zebra, like the zebra, the animal, chips. Okay? And in the US, people love to eat chips, right? But they like their chips to be like this. So chips like that get rejected. Yes?
Does it taste bad?
No. No.
Like a new variety.
Specialty,
Like,
yeah.
Yeah, but people don't like it. people do not like it. And they say that it has acrylamide, so it's not so healthy, okay? But so what happened is that from the moment that this disease arrived, I'll show you the map, it's really, really close to Texas. Very quickly the disease was found in Texas, okay? So, but when I think potatoes in the US, we don't think Texas, right? Which states do you guys think? Idaho, okay. Because Idaho, Oregon, and Washington, they produce more than 50% of the potatoes. So why do we care in Texas? But because in Texas, we have a very nice potato industry. 70% of the potatoes go to the chipping industry. So in fact, Frito-Lay, who makes Lay's chips, the headquarters are in Dallas. Okay, so Texas chips are important. So this disease that arrived very quickly to Texas, immediately the growers had the potatoes that were being rejected, and that cost a lot of money. So very quickly there was an effort to try to understand this disease. I'm talking early 2000s, 2003, 4, 5, okay? And so from its initial description in this unique potato field in Mexico, the disease expanded, okay? And it went, it went south to Mexico, reached Central America, and starts going north until it reached the Pacific Northwest, where more than 50% of the potatoes are produced, around 2011. And you can see also that it arrived to New Zealand, okay? And that was a shipment of plants that was infested. Now we know there was a vector, and the vector had the pathogen. So, but very early, the disease arrived to New Zealand. So, but as I said, there was an effort to try to understand the disease. And so, very quickly, 2006, it was identified the disease was associated with a vector, the potato psyllid, also known as the tomato psyllid, Bacterial sarcocarelli, is a phloem feeding insect, native from the area, southern US, northern Mexico, known by the growers forever, never a major pest. Then it was found that the causative agent of the disease is a bacterium. The name is here, Candidatus Larybacter solanacearum, very long, so I'm going to be calling it LSO from now on. And it's a gram-negative phloem restricted bacterium that is closely related to another pathogen that is famous in the US. Somebody heard of any Larybacter pathogen? Citrus greening, very good. So it's a relative to citrus greening, which now we call Huanglong Bing, but that's okay. So the agent is Candidatus Larybacter asiaticus, and this disease is so important that it deserves being mentioned in the New York Times, Los Angeles Times. So it's a big deal. This costing millions and millions of dollars to the citrus industry. But we're working so in potato or tomato, and our pathogen it infects solanaceous plants. So solanaceous potatoes. Any other solanaceous? Tomatoes, peppers, tomatillos. So if you live in Texas, a lot of those we eat. So this is the pathosystem that we are going to be working with. Okay. I told you that when we have a disease, ideally, if we can find host plant resistance, we are safe because there's going to be no disease in spite of the fact that you have vectors. In spite of the fact that you have the pathogens, there's going to be no disease. So very quickly we team up with the potato breeder in Texas A&M to start doing testing varieties to see if we could find resistance. And we took advantage of the fact that in Texas it's so big that we have two different growing areas in Texas for potatoes. We have the lower Rio Grande Valley, here in the south, in the border with Mexico, and then we have the Panhandle here. Okay, and so they are so different, so in altitude and in latitude, that in the south we plant around November, December, and we harvest around April, whereas in the north we plant around April and we harvest around August, September. So complete different environments allow us to have 2 seasons per year, test a lot of material. Here with this beautiful star, I mark College Station where Texas A&M is located. So this is 6 hours drive. This is more than 10. People from the Panhandle. Depends how fast you drive, right? So it's far, far, far. And the way we do that is we will go to the growers' fields and we will plant our plants inside these tents. And when the plants grew, we will infest each plant individually with our psillates, and we only allow them 1 week to feed, and then we can come and collect all these bags. You guys cannot see the mouse? Can you see the mouse, right?
Yes.
We will collect the bags, and so we can collect our insects, so we are not leaving insects in the field. And the tents, the role is to protect our plants from the natural psillates, so our negative controls are still negative. And in case we have any escapees, well, the grower is protected. And so we tested varieties, we tested selection, we tested advanced selections, we tested relative species, and there's no resistance that we could find that was of interest. So you need to have resistance, and it has to be commercially adapted. So nothing, we couldn't, and we were not the only ones looking for that. So no resistance. We have some tolerance, but not very much. So coming back to our disease triangle, ideally, we want to break this interaction, right? And so the plant resistance is a very good way to go. We couldn't, okay? And so then the idea is, okay, let's try to understand how the bacterium infects the plant. If natural resistance doesn't occur, can we make it happen? So we need to study the bacterium. The problem that we have is that our bacterium is non-culturable. So it's very difficult for us to work on the lab. And and so we have to find ways to try to protect our plants. So since these plant resistance fail, so we can focus on the bacterial pathogenicity. Another idea would be, can we disrupt the transmission, or we can disrupt the feeding of the insect on the plant. And this one we can use maybe antixenosis, antibiotics that is typically used, okay? So this is why my talk is a triptych, because we're looking at all aspects of the interactions to try to disrupt it. And so while we were doing all these, in fact, the situation kept advancing and kept changing. This is a more or less today map, more or less. There's things that are mistake here, and I'm going to go through this. So something that happened is that very early on, when the disease, we were all trying to figure out something about the disease, we will have annual meetings where we will have scientists from the US and Mexico, as well as scientists from New Zealand, because they wanted to try to stop the disease. And so we were talking about our results, and all the time the scientists from New Zealand would say things like, well, in New Zealand the disease is not like that. Well, in New Zealand the disease is not like that. And because it was invasive, right, and it came from the US, we thought that, well, maybe it was the potato varieties that were different, maybe the climate, the soils, who knows. So there was a lot of exchange of materials planting New Zealand varieties in the US and US varieties over there to try to find what why we had those differences in the diseases. And kind of everything came a little bit into context when we realized that there were different haplotypes of this bacterium, genetic variants, and that in New Zealand, only haplotype A had arrived, whereas in the US, we had two haplotypes, A and B. And all the scientific community was sharing the same materials, and our colonies were infected at the same time with A and B. And in spite of the fact that our colonies were double infected, sometimes we will have transmission of A, sometimes transmission of B, sometimes of both bacteria. So maybe some of this noise was due to this double infection. So as I said, the situation was changed. I cannot make a map with only half of the US is marked, so there's no zebra chip in the eastern half of the US. It arrived to Canada, but obviously it's not all Canada. It's not in Alaska, okay? Yeah, it's a mistake. We have the pathogen that is found in Europe, but there's different haplotypes. C, D, and E, they have a geographical distribution, but they are associated with carrots, Epiaceous crops, and caropsyllates. U is urticaria, so it's a weed. in the US, we have now other haplotypes that are not as well characterized as A and B. We have over 10 haplotypes of this pathogen, I don't know how many, more than 10 that exist. And very, very recently, the disease arrived to Ecuador and Peru, and I think Colombia, and I think Brazil. And you know, Peru is the center of origin of potatoes, so it's a big deal that the disease has arrived there. But so we in the US, we focus in A and B. And so the questions that we had is, okay, we do not know anything about the disease. It's very difficult to work because the bacterium is unculturable. So we thought maybe we can use this genetic diversity to try to understand something about this disease. And so the first idea was, well, maybe the difference between New Zealand and the US are linked to these different haplotypes. So let's separate the haplotypes. It's easy, but it took us time. We separated the haplotypes, and then we start asking questions as like, do they cause the same diseases? And so I'm going to show the results for tomato. We work a lot with tomato in the lab. So here we have control plants. It means they are not infected. And here they are LSOB and LSOA infected tomato plants. This is 6 weeks after infection. So you can see that the infected plants, they are stunted. From far away, you don't see any other symptoms, but there's some deformations on the leaves. But overall, A and B look pretty much the same, right? Yeah?
It's infecting just the plants, right?
No, it moves throughout the plant. It infects everything. But it eats a lot in the root, indeed. We look at the same plants 3 weeks later. This is what we see. All LSOB infected plants are dead. LSOA infected plants, they are not dead. They are stunted, they have some deformations of the leaves, but they are alive. And we kept them 14 months, and they were alive. So difference in virulence associated with tomatoes. So it's interesting for us, because now we can start comparing haplotypes, right? We did the same, so the same studies. This is in tomato. In tobacco, we found the same differences. In potato, B is also more virulent, but both haplotypes causes plant death, okay? So there's some differences, but it appears that LSOA is less virulent than LSOB. And so then we ask the question, what about on the vector? Okay, previously we had studied the effect of LSO infection on the vector before we knew that we had double infected colonies, and we knew that the presence of the pathogen reduced the fitness of the insect. So it's a pathogen also of the insect. So now we repeated the same experiments, and I'm only showing here the results of infection on oviposition and nymphal survival. How do you make the arrow to become big? I don't know. Suddenly the arrow is huge. I don't know. So, and we see that both LSO and LSOB separated, or when they are together, they have they make the females to lay fewer eggs, and they also affect the nymphal survival. So both of them have a fitness cause, but again, LSOB is more pathogenic. So we have LSOB is more pathogenic than LSOA. And so the question that then we had, okay, what about, can we use this information to disrupt somehow transmission? Because in fact, LSO is a good biological control of the colonies. Our infected colonies are more difficult to maintain because of the fitness cause. And we were like, okay, if we can understand what's going on, it's not that we're going to release LSO there, but maybe we understand something about the biology of the insect and the biology of the pathogen. So we set up to, let's try to see if we can disrupt transmission. So let's study a little bit the interaction between the vector and the pathogen. And so this is work of a former student, Xiaotian Tang, who graduated like 3 years ago already. And the question is, how does Bacillus defend against LSO? Okay? And so what we know is that LSO is transmitted in a persistent, propagative, and circulative manner. All this means that once it's acquired, the insect remains infected the rest of its life. Propagative, that the bacterium replicates inside the body of the insect, and circulative, that it has to go through the gut and the salivary gland, it circulates, okay? And so the question that we asked is whether there were differences in acquisition and transmission of each haplotype. So what Xiaotian did is he took adults, uninfected adults, and allowed the insects to feed by 2, 4, 6, 8 days, etc. he dissected the gut and he measured the amount of Libribacter in the gut of the insects, A or B separated. And so in blue, you have the titer of LSOB in the gut, and in red, you have LSOA. This scale is log 10, so it seems like there's no major difference, but there are differences in there, okay? And what you can see is very quickly, the titer of LSOB increases very quickly, and by day 6, it reached the plateau. You see the little letter E everywhere here, okay, plateau. Whereas LSOA, yes, it increases the first, it's acquiring, but the increase is lower, and you see the letter E appear for the first time here at day 12. So there's 7 days more or less, it's a little bit less, but a week of delay on the accumulation of LSOA with LSOB. And so the question was whether that will, this is acquisition, will make a difference for transmission. So what Chaltian did is he took the insects and he allowed them to feed 7 days, and then he, every 4 days or so, he would put them in uninfected plants, switch them to new uninfected plants, and then we see when the plants first become infected. And we see that first become infected around the day 21. This is the first day that we didn't test every day, we test every 4 days or so. And so, but we see that when the first infected plants appear, there's many more plants that are infected with LSOB, and the amount increases very, very fast, whereas LSOA, it's very slowly. And so probably that means that indeed this difference in acquisition also translates in a difference of transmission. LSOA is taking probably longer to circulate in the body of the insect and then be effectively transmitted into the new plants. So it appears that whatever is happening in the gut is important. The gut is delayed in the infection with LSOA, and this delay infection has an effect on the transmission. So we were really interested in what is happening in the gut, in part also because the gut is the first organ that gets infected, so it's easier for us to work with, whereas it's a lesser organ, you want to wait until the insect circulates, and we cannot see very well when that happens. So we decided to focus on the gut, and so what are the responses of the gut to Libribacter? So Libribacter is an intracellular pathogen, and a very good defense of eukaryotes against intracellular pathogens is programmed cell death. If you kill the cell, you're killing the pathogen, you're stopping the spread of the pathogen. And in the citrus system, they had said that in the gut of adults infected with a pathogen, there was apoptosis. Apoptosis is a programmed cell death. So we said, okay, let's look to see if we see apoptosis in the gut of our insects in response to either LSOA and LSOB. Is typically a response of eukaryotes. So we looked and we looked, I forgot that, to how we do that. In all these experiments, I'm going to be comparing LSO-free psyllids, so no Libribacter, LSOA or LSOB infected colonies. They were infected all their lives since nymphs. And then we have adults that we infect at different time points. So we look at 2 days after infection or 7 days after infection. So we know that it's early in the infection or later. Remember this increasing titer in the gut, right? And then we dissect the gut and we see whatever we are looking. So we look for this apoptosis in the gut, and we look in insects that were infected since they were nymphs, insects that just acquire, insects that acquire a little bit later. And we did all these different measures, different ways to see if you see apoptosis. And we look and we look and we look, and we found no evidence of apoptosis in the gut of psyllids in response to either LSOA or LSOB. And on one side, we said, it's okay. Different systems, different responses. But on the other side, we are like, why there's no apoptosis? Because apoptosis is the mechanism that should be deployed to stop these pathogens. So we decided to look at the gene expression in the gut, looking for the expression of genes involved in apoptosis. And what we found is that in response to LSOA infection, so this is before infections, 2 days, after infections, 3 days, 5 days, 7 days, we see an induction, upregulation of an inhibitor of apoptosis. This gene inhibits apoptosis. And at the same time, there is a downregulation of a caspase. Caspase is the executor of apoptosis. So caspase is making apoptosis, and the inhibitor of apoptosis is in fact inhibiting the caspase. So we see that caspases are downregulated and the inhibitors of apoptosis are upregulated, so no apoptosis. This is in response to LSOA, and the same thing in response to LSOB. So this seems like a common mechanism, and we didn't see apoptosis in response to either LSOA or LSOB. Okay, so we don't know what it is, but we decided that maybe in that case, it was worthy for us to try to see what was happening here. So what we decided to do was to silence this gene. So we silence the inhibitor of apoptosis here in gray. Is the silenced insects in the gut compared to the non-silenced insects gut expression. And we see that in parallel to the silencing, there's also an upregulation of caspases. So there's a link between these two that is not known to be linked, but we found it. And so we measure and to see if insects that were silenced for apoptosis, they have difference in titer of apoptosis. Okay? And indeed we see that the insects that are silenced for the inhibitor of apoptosis, so they are developing apoptosis, the titer is decreased. I don't know if it makes sense. Yes? Yes? And so once we silence this inhibitor of apoptosis and we see that we can reduce the titer of LSO, we decided to look to see if that would affect acquisition and transmission. So the experimental setup that we did is we have insects, uninfected insects. First we infect them, then we induce we induce silencing by RNAi, and then we transfer the insects to uninfected plants regularly to see if we can decrease transmission. We first infect the insects so that the feeding is not affected by the apoptosis or the RNAi. So to make sure that they will have opportunities to feed and acquire the pathogen as much as they could. And here's what we see. In blue, you have the insects that were fed double-stranded RNAs, so no silencing is our control. And in red is the insects that were fed the double-stranded RNA, so where RNAi is happening. And here is the percentage of infected plants. So we see that insects that were silenced for the inhibitor of apoptosis, so where apoptosis was happening, where Libribacter accumulated at lower levels, then they transmit less bacterium. So everything is working very, very nice. But we don't know the mechanism, okay? This is where we are at. We don't know the mechanism, and this is the model that we have. What we think is happening. What we think is happening is this is the gut of the insect. LSO arrives through the lumen, is going to enter in the gut cells. The gut cells are wanting to respond by apoptosis by Libribacter is disrupting this response, and as a consequence it can replicate, cross into the hemocell and distribute itself. So how does this inhibition of apoptosis occur? We do not know. But pathogens, what they often do is they secrete these effectors, so there are proteins that they secrete, have an effect on the host, and part of those effects are disrupting immunity. And so we marked these potential effectors are these little color dots. Those are putative effectors, different effectors, and one of them are going to be involved in stopping apoptosis, okay? And this has been shown in other systems. So this is what we are looking for. We have also recently published the transcriptome of the gut of the facility in response to A and B. I'm not going to go into detail, but we have overview of other potential responses that may be the difference between the response to LSOA and LSOB, because apoptosis is common to both of them, okay? So if you want to go and read the papers, go ahead. And so coming back to my triangle, here I wrote that we investigated and we saw the potential role of apoptosis on the interaction with between the pathogen and the insect vector, and that we showed that we were able to reduce transmission by manipulating the system. Okay, we don't know how. Okay, no, we are interested in how we can do that, and so we are interested in knowing how does Libribacter attack the plant and the insect, because we know that Liberibacter can disrupt the immunity of the insect, and I didn't show you, but we know that it can also disrupt the immunity of the plant. So how does it do it? And I told you that pathogens use effectors to do so. So now we are looking for LSO effectors. So here I have a little diagram showing what's an effector. Effectors are these blue balls. Those are proteins that are produced by the pathogens, and they are secreted directly into the host cell, and those effectors will interact with different proteins, and they have the ability to stop defenses. Okay, because a defense is an arms race, the host can also evolve mechanisms to detect those effectors and stop the effectors. So it's a constant battle. Okay, but we're interested to know what are the effectors that Liberibacter produces. So how we identify effectors? Effectors are secreted proteins, and so they have this beautiful N-terminal domain that is very easily recognizable in by bioinformatics. So here's a protein, and this is the prediction of secretion domain, and this green dashed line shows that this domain is going to be cleaved, and so the effector, the protein after being secreted, is liberated in the medium. So that's a perfect example of an effector. So we took LSO genomes and we looked for potentially secreted protein, and we have more than 100 putative effectors. Some of them that are common to LSO and LSOB, and some of them that are unique, and some of them that are slightly different. And so now we want to see among the 100 putative effectors, which ones are really effectors and which ones are the ones that are really manipulating the immunity. And so we set up a pipeline where we run our effectors through. And so the first thing that we do is we evaluate to see if there's difference between LSO and LSOB for these effectors. We then test the secretion. It's not because the computer tells us that it's going to be secreted that the protein is really secreted. We cannot culture Liberibacter, so our secretion test is done in Escherichia coli. And what we do is we do 3 steps. So first we clone the signal peptide of the effector, the one who says this protein needs to be secreted and liberated in the outside. We clone it in frame with a phosphatase alkaline gene of Escherichia coli. If this gene gets secreted, the bacteria will turn blue. Okay, and so here we have a few signal peptides that drove secretion. One didn't drove secretion, and the other one is a negative control to make sure that our system is working. So we go, and this is the first screening that we do. Can the protein be secreted? It's not because the bacteria turned blue that the protein was liberated into the outside medium. It's just it was exposed. So the next test is to see whether they are really liberated. So what we do is we grow the bacteria, we collect the supernatant, we filter sterilize the supernatant, and then we put the reactive. So if the medium turns blue, it means that the protein was outside. And so here we have an example of the supernatant of one of these bacteria that turned blue, meaning the protein was there in the supernatant because there's no cells whatsoever in these tubes. Okay? And here we are only testing the signal peptide. So now the next step, the ones who pass this secretion test, we clone the whole full length gene and only with a his tag, so it's a smaller tag. And we do the same thing, we grow the bacteria, we filter the bacteria, and we test whether the protein was present using the tag in the cell and or in the supernatant. And here we have a few examples. So here is the cells, and we see here, I mark with the asterisk the expected size of the protein, and we see that those proteins were indeed in the medium. So those were secreted, okay? And we have the first one here that was not secreted. This is one that was white in this test. And we have this one here that is also not secreted. This one is one that passed this test but failed this test. So we have a very good correlation of things working. So we are pretty confident that they are going to be secreted. And so the majority of the proteins pass the secretion test because this sec domain is very well studied, predicted, so the computers know how to identify it. It's just in case that we do it. And the proteins that pass the secretion test, we move them forward in our pipeline. And so the next things that we looked is whether they are preferentially expressed on the bacteria when it's associated with the insect or with the plant. And then we are interested in the ones that can suppress immunity. So we have different bioassays that we develop. We test to see if they can suppress programmed cell death, that I told you is a very nice way of disrupting the infection by intracellular pathogens. Whether they can disrupt ROS signaling or calcium signaling, those are different steps in the signaling of infection. And so I'm not going to present all this data, I'm just going to present how we test for programmed cell death. So we use transient expression in Nicotiana benthamiana, and we use a gene that is PRF. PRF is an immune gene that when the plant detects a pathogen, activates this protein, conformational change, and there's signaling to activate defenses. So PRF-DV is a mutant that is always active. So it's active without detecting a pathogen. You express it in the plant, and you have cell death. So we just infiltrate an area of a leaf, and the area that is infiltrated dies. Okay? It's a very well-known system. It was developed to test effectors. And so what you do, then you co-infiltrate with an effector that is very well studied. And for example, you see that this effector is able to disrupt the cell death. Okay, that is our positive and negative controls that we use. And then we co-infiltrate our different candidates with PRF, and we find the ones that are able to disrupt programmed cell death. When they are able to disrupt programmed cell death, it means that they are able to disrupt somewhere on the signaling cascade. We don't know at which step, okay? And we do have similar things to find which ones can disrupt ROS, which ones can disrupt calcium. And so we have now a list of candidates that can disrupt immunity. And so then the next step is, okay, how do they do so? And so we have tomato and psyllid hybrid libraries that then we screen with our effectors to try to identify the partners in the host to see how do they function, these effectors function. So I'm very quickly going to show you the results for the first effector that we characterized, that we call HPE1, hypothetical protein effector 1, because the majority of those effectors have no similarity to anything that is known, so they are all hypothetical proteins. So very difficult for us just identifying them, knowing what they were going to do. We need to go through all the process with all of them. So HPE1 is a protein of unknown function. It's a secreted protein based on our assays. It's expressed at the same time in insects and in plants, so it may be involved in disrupting immunity in both organisms. Can very efficiently suppress PRF-DV cell death, cannot suppress ROS signaling, cannot suppress calcium signaling, and it was, as I said, very effective in disrupting this cell death. So it was the first effector that we moved through our pipeline, was so efficient disrupting cell death that we move it forward to the yeast hybrid part. So we screen our tomato yeast hybrid library, and we fish out a RAT23 protein. Here you have the results of the yeast hybrid library. What screening, what you need to see is that when we express our effector and the tomato RAT23 gene, the yeast grow in a selective medium. This means there is interaction. The controls do not grow, and this is a positive control. Okay, if you have growth in here, it means the proteins are interacting in yeast hybrid. We did other verifications to verify that indeed this interaction occurs. And so what is RAT23? RAT23 is a protein that is involved in shuttling ubiquitinated proteins for degradation into the proteasome. Okay, very important to protein turnover, which is essential for immunity. This protein has kind of two parts. In the N-terminal side, it has this domain, the UBL domain, that interacts with the proteasome. On the C-terminal, it has the UBA domains that interact with the ubiquitination of the protein. So this part of the protein binds to ubiquitinated proteins, and they take the proteins to the proteasome, and this part give the protein to the proteasome for destruction. We know that our effector interacts, I lose the mouse, with this part of the protein. So based on that, we hypothesize that our effector is going to disrupt the degradation of ubiquitinated proteins, So we were going to have accumulation of ubiquitinated proteins in our plants. And so we tested that. What we did is we expressed our effector, this side, and here is the effector is not expressed. Here I'm showing you that the effector is being expressed in these plants, where it's not expressed in these plants. And we looked for accumulation of ubiquitinated proteins just using an anti-ubiquitin antibody. And we can see that 36 hours after infiltration, so the protein is starting to be produced, there's starting to be accumulation of these ubiquitinated proteins. So indeed, that is what our effector is doing. How that is helping infection, we don't know yet, okay? But based on that, we decided to test our infected plants, and infected plants also accumulate ubiquitinated proteins. And we also created transgenic plants that express this effector, and we see that those infected plants become infected faster. So the titer of LSO increases faster and the symptoms develop faster. So it's a virulence factor, okay? This expression really can dampen down the ability of the plant to get infected. So here is what I just said that we have shown. And also we have shown that in the citrus system, there is a homolog effector that has a similar function. So we can take our results from tomato and move it around to other systems where these proteins exist, which is only Liberibacter so far. And so if we come back to my disease triangle here, our screenings allow us to identify HP1 as a virulence factor and RAD23 maybe as a susceptibility factor. It's difficult to see. And that when we express HP1, we have the infection that was enhanced. So we can reduce transmission, we can enhance infection. The question is, can we play with these systems now to maybe avoid infection, okay? I'm running out of time, so I'm going to go very quickly. I think I'm running out of time, right? I haven't finished yet,
It's
so.
okay. We don't mind.
Okay, because it's a class, right?
They don't mind.
Okay, so that's fine. So I'm going to go through it. And so I want to present the third part of my triptych, that is the interaction between the insect and the plant. Can we disrupt this? Can we disrupt feeding? Okay? And so psyllids are phloem-feeding insects. Everybody knows how phloem-feeding insects feed? Everybody knows this diagram? Perfect. So the way phloem feeders feed is they use their stylet and they go intercellularly until they reach the sieve element, and at that moment they are going to take up the phloem, okay? But this feeding is possible because all along the path the insect is constantly salivating, and there's different saliva that are produced, and also throughout the path the insect is probing these different cells, and what is that is do a little salivation and then taste, and this tasting allows the insect to perceive the defense of the host, the quality of the host, how good is the host, and also to find when they found the phloem, because you cannot see. So the difference in composition say, oh, here is the phloem, okay? And so there's different salivas that are produced throughout this process, and those different salivas are marked in red, for example, in blue, and in pink. And these salivas are different, and they have different functions. For example, the one that is in red protects the stylet mechanically from the abrasion of the plants, also from the defenses that are present there. The ones that are the ones in the probing also needs to disrupt the plant defenses, because the plant will defend itself. And then the ones in the phloem really has to stop the phloem from shutting down. A plant that is detecting an infestation, the first thing that it's going to do is going to stop phloem flow in those cells. Here, in the sieve plate between sieve elements, there's going to be deployment of P proteins and then callose deposition. And the result is that the phloem doesn't flow anymore. If phloem doesn't flow, the insect cannot feed, the insect goes away, and after a while the plant resumes phloem flow, okay? But it's a very effective way. So if the insect needs to feed effectively, it needs to be able to disrupt all those defenses of the plant, okay? And here I have sequential events of type of defenses that may occur in response to insect feeding or to pathogen infection. Is the same. And here we have things like the calcium that I talk about, the ROS, changes in in hormone could be JASA depending on the system, gene expression and expression of defense proteins. So the insect, just as the pathogen, needs to be able to disrupt all this in order to feed efficiently. Okay.
Can I just ask a question? How long does it take? I mean, do they feed for a long time?
Yes, if it depends on the quality of the host, how long does it take to reach this level, and once they reach the phloem, the first thing that they do is salivate a lot, and this salivation kind of stops the defenses, and if the host is very susceptible, that is a short period of time, and then they can ingest a long time. The more defenses the plant has, the more they salivate, and sometimes they will salivate without ingesting. They cannot reach ingestion and they will leave, but there will be a lot of salivation. It could be hours. Okay, and so we have set up this beautiful pipeline to test the bacterium effectors. Insects produce salivary effectors, so now we're using our same pipeline to evaluate what are the proteins that the insect produces that allows them to manipulate plant defenses. And we have two strategies. The first strategy is to use a candidate approach. Which proteins have been identified in other phloem-feeding insects such as aphids, CO2, or whiteflies, BSP9? Well, those are specific. We have none of those in psyllids. Then we have others that are more conserved, like peroxidase and stuff like that. Those ones we have identified. The second approach that we have taken is to take a salivary gland transcriptome to identify the genes that are expressed in the transcriptome, identify of those which ones are predicted to be secreted, and then we move them through the pipeline. So we have more than 100 genes that are expressed in the salivary glands, 35 of those that are predicted to be secreted. And we have things that are known, like pectinases, so they're going to be involved in disrupting the cell wall, and many others are of unknown function. Those are the ones that I'm most interested in, the ones that we don't know what they do. And then we move them through the through our pipeline, okay? We haven't published any of those, so I'm not going to give a lot of details. But so we evaluate the expression, we look to see whether they're conserved, are they unique to psyllids, are they unique to phloem feeders, are they unique to Bacterial Sarcocarelli. We look at the role in suppressing programmed cell death, we look at the role in suppressing ROS, calcium, I'm blanking, the callose deposition, different tests, and we see which one they're interested. The ones that are interested, we look, we screen our isohybrid library, and for example, the first one that we did that very efficiently suppresses cell death and very efficiently suppresses ROS, we're screening our isohybrid library. We identify an interaction with a protein that is involved in binding callose and working on opening and closing plasmodesmata, which is very important for phloem flow. So I'm not going to talk anything else about that because we have not published and we are far from publishing, but we are going. And so this is kind of my last slide, and it's because the system was already not complex enough. If you guys like complexity, we can always make it a little bit more complex, and it's just for you guys to know that in fact I talk about 2 different haplotypes of the pathogen, and there's others, so we work with 2. But we also have different haplotypes of the insect, right? And so at least 4, the northwestern, the central, the western, the southwestern. Here is Venn diagrams in Colorado and surrounding states. And the different parts of this pie diagram represents how many insects of each haplotypes you can collect. So you can collect all of them at the same time. And those haplotypes have a somewhat geographical distribution, somewhat. They also have different microbiomes for people who like microbiomes. They have different plant usage. They may have different transmission. So we can keep adding levels of complexity, but we are working very simple. One haplotype is more than enough for us. But there's more to be done with this system. And so after talking almost an hour, what are the conclusions that we can draw from our work? Okay. So the things that I'm happy about is that we have identified proteins that appear to be involved in the interaction between all these organisms. That are the things that I like. How do they talk to each other at the molecular level? We have shown that we can disrupt these processes to disrupt transmission. We have also identified kind of susceptibility factors in plants. So the question is, can we now use that information to make resistance in those plants? I don't know. We have previously shown, and I didn't talk about that, that only antibiotics and anticyanosis against the insect will not protect the plant, okay? The insects are not going to feed a long time, they're not going to reproduce a lot, but there will be transmission, okay? That doesn't protect against the disease. Can help in an IPM strategy, ways to fight against the vector. But in spite of all the advancements that we have made, and for me they're huge advancements, we still have lots of questions about the mechanism, how to put everything together, and more important, once we get this knowledge of how the infection occurs or the transmission occurs, how can we leverage that knowledge to use it effectively in the field? How can we deploy things in the field? If we find ways to make plant resistance, that is, today we can make plant resistance, but how long is the resistance going to last? If we identify ways to make the insects not transmit the pathogen, how do we use that in the field? Those are the questions that we need to be focusing on, that we are not focusing enough, and the system is challenging. So to finish, I would like to thank current members of the lab, and I talk about Azucena's work, former members of the lab. So Azucena is my technician. She's been with me forever and ever, and she's involved in everything. She's, Chloe can attest, I say she's the boss of the lab. What she says goes. Forrest Chachan-Khan is the one who worked in the effector and the tomato plants. I talk a little bit about Punya's work, which was the effect of the haplotypes on the psyllid fitness. I talk about Shatyan's work, that is the interaction between the psyllid and the bacterium at the gut. I want to say, acknowledge my collaborators. So Rodney Cooper and Gina Angelella, they are ARS, and they are my collaborators on the work on the salivary glands. So this is the newest part of our work. Murad Ghanim in Israel is my collaborator in the part insect bacterium interaction. And Julian Levy, he's also my all the time collaborator, and he's involved in all the projects I talked there, and the ones that I have not talked, and funding sources. And so before I shut my mouth, if you guys are interested in anybody here in coming to work with us, we have open positions, so don't be shy. Texas is a wonderful state to live and to work. It's warm. We have 95 degrees outside at this time of the year. So we live in summer. We have winter also, don't worry. So you get your few days of cold, and then we enjoy the heat again. So please let me know if you are looking for postdoc positions or technician positions, or if you know somebody who is interested. Thank you very much for your attention.